Good morning, gents. This is a quick video explaining the Gene Lab assignment. So here we go. So basically with your assignment, you'll get your task sheet, of course. It's important to read through this. So basically you're a part of a team of vets that works with Dog Breeders Association of Australia as a genetic counsellor. So that's who you are, and you're writing this report from that perspective. Your client has a Labrador bitch that has a family history of progressive retinal atrophy, a condition which causes gradual blindness. So first of all, you need to go and research what that is. Okay, find out information about the PRA, how it's caused, any genetic information, whether it's dominant, recessive, co-dominant, or anything else. All right. The reason why you want to do that is because your client wants to breed the dog with three sires. So the female bitch you've got a choice of three male sires, and your job as a genetic counsellor is to determine which of those male dogs is best to breed with the female in order to produce puppies with the least chance of PRA. Okay, so you're going to have to use pedigree charts and Punnett squares to be able to work out which is the best combination of male dog and female dog to be able to produce puppies. And you need to do that by analysing their particular genetic makeup, their history, to work out all those unique little things there. Okay. There are three available stud dogs, as I said. Given the pedigree of each of these three dogs, you must determine which of them should be selected for the litter. You'll give your recommendation to the client in the form of a genetic report explaining your decision. You need to include family trees, mathematical probability of the phenotype and genotype. Okay, so not only whether it does or doesn't show PRA, or whether you also need to show whether what their genotype is. And you need to, at the end of it, you actually have to make a recommendation. Say that, it is the recommendation of this report that this male dog be paired with the breeder's female dog. Okay, it's important that you justify both why you do recommend one dog and why the other two are not suitable. So don't just simply go, I picked dog A. You need to explain. If you're going to pick dog A, you need to explain why B and C are not suitable. Okay, so looking through here, this is all stock standard. Fill these in, read through that, sign your assessment policy document. And then finally, this is the final page of this. I'll get another document in a minute. Okay, so it's important that you read through here. So obviously I would be hoping that all of you are looking in this category here, okay, looking at going, what do I need to do? Okay, so first of all, my science understanding. You'll know this in a minute, but essentially what I'm looking at is parts 1, 2, and 3. What is PRA? The introduction, what are recessive and dominant genes, and a description of PRA. Okay, so here's where you're doing that. A clear description of all theories underlying the mechanisms of genetics and inheritance, including dominant and recessive, expression of genotype and phenotype, and the biology of PRA. So that's what you're looking to do. Okay, scientifically justified explanation of stud male recommendation using accurate analysis. So that's later on. That's part 5. Part 5. Okay, when we go up here, the science inquiring skill. This is your calculations and your determination. So you need to show how you ended up with your recommendations. Okay, needs to be accurate calculations. You need justified explanations. Okay, you need to accurately manipulate and analyze the Punnett squares in order to explain what you're doing. Okay, so have a read through these because this is what you're aiming for, and it very clearly steps out what you need to do in order to achieve an A or a B or a C. Okay, moving on now. This is a report template that you'll be given online. Okay, but before we do that, right at the start, we're going to actually watch a video on it. Okay, so you need to, in order to do that, you need to come to Jack Plus, go to chapter 2. All right. If you click on, well, it'll come to here. If you click on this button here, resources, and go down to Project Plus, you'll see this thing called the Gene Lab. Okay, so click on that. It'll take you to a new page. Okay, and then watch the video and have a read through here. Okay, it's important you do that. We'll be doing this in the first lesson in my class. Okay, but you can always go back to this. So again, whoops. Close that. go to Chapter 2, go up to Resources, then to Project Plus, and there it is there. Okay, so have a look through that. So back to here. This is your task. This is a repeat of what's gone on, so no need to read through that. Okay, this is your front cover. You can pretty that up a bit, name, class, all that sort of stuff. Insert a photo. You get your index sheet here. Do this at the end. So once you've done everything, go back and do this so you can get the page numbers correct. Okay. Please delete this. Okay. This is instructions. I don't need to see this again on your report. So please, anywhere where there's instructions, they need to be deleted completely. Okay. Your introduction. Okay. It's a brief summary of the purpose of this report. Okay. So it's not about I, we, me, or our. It's the purpose of this report is to do this. It is at the request of who, okay, and at the end it aims to do what. Okay, so do that. It says 50 words. That's a guide. Okay, please don't think you have to keep to 50 words. I would expect that most, if not all, people are well over 50 to be able to give an accurate introduction. However, I don't want 500 words. Okay, you know, under 100, 200 words is fine as long as you get the basic information in there. Also, you need to make sure you include in-text referencing. Cite your sources. Okay, and I'll talk about a bit more about that in the end. Recessive and dominant genes, you need to talk about these, what they are, how they work together, you know, how the genes are passed from parent to offspring, what are dominant recessive genes and how they express in the phenotype. Okay, you can use diagrams, illustrations, tables. Again, the 50 words is a guide to tell you that you shouldn't be going to 500-600 words on this. Keep it short, but make sure you include all the information. Don't feel like you have to stop at 50 words. If you have to say it in 100 or 200 words, that's fine. Okay, and finally, description of PRA and looking at these questions. So what is PRA? What is its effect? Can it be treated or cured? Okay, so make sure you get the distinction between treating symptoms and actually curing a disease. Okay, so treating a symptom is actually making the symptoms better, but you've still got it. Okay, whereas curing it means that you completely get rid of the disease. Okay, so is that possible with a genetic disorder? Okay, ask yourself. So be careful about how you use your terminology when you're talking about treating or curing, if you're going down that path, but make sure you talk about either. So can it be treated? Yes or no? How? Can it be cured? Yes or no? And how? How common is it? And is PRA a recessive or dominant gene? Okay, and again, once you've started, get rid of this section here. Delete these questions. These are for your help. I don't need to see them again. Okay, finally, once you've done that, we need to go, you've done your introduction, your background, and you know what PAR is. We need to analyse our pedigrees. Okay, you will get a third document from your teacher, which gives you a particular pedigree chart for the bitch, the female dog, and the size, the 3 male dogs. Okay, and then you have to analyze these. I won't show you these now because we'll start the first 3 sections first before we get into it. But this section involves looking at them. Okay, none of the dogs at this stage show any sign of the PRA. Okay, what does that mean about them? Okay, they're obviously carriers. Okay, but some will be, you look at their heritage, so you'll be looking at the dog in question. You'll have his parents to look at and their phenotype, and essentially their grandparents and their phenotype. Okay, so you use the description of the dog's predecessors, so its ancestry, to determine what its possible genotype is. Okay, in some cases it's going to be very clear, others not so much. Okay, and then we want to go through here. Again, get rid of all this when you're doing it. Okay, so when you're analysing your pedigrees, you'll be able to draw on them, do Punnett charts. We need to see those. You need to show your calculations. So, for instance, if you do your Punnett chart and you work out its 3-to-1 ratio, what does that mean? What does that mean in terms of percentage? Okay, when you cross them over against the male versus female, where are the percentages? What is the chance of the puppy being homozygous dominant, heterozygous dominant, or homozygous recessive? Okay, so you need to work those out and present the maths accurately. And finally, we do our predicted results of breeding. So section 4 is primarily looking at the adults, okay, and looking at the bitch and the 4 sires and going, this is their this is their particular genotype or possible genotypes. Okay, section 5 is where you cross them over and work out with the results of the breeding. Okay, so you need to determine the genotypes of the Labrador and the mates, cross them over and work out, okay, if I cross the bitch with this particular SCI A, these are the chances. Her with SCI B, here are the chances. Her with SCI C, here are the chances. Okay, and outline them very clearly. Okay, you can read through this carefully, and again, get rid of all this before you print it off and hand it in. Okay, so with this, please make sure that you're referring back to your pedigree charts and Punnett squares, okay, and using the figures you've shown in your calculations, okay. When you make your predictions, okay, you also want to make your recommendations as well. So finally, as mentioned earlier, has been based on the Punnett, the calculations provided. The female with SCI A or SCI B or SCI C is recommended because of these reasons, as demonstrated by this and this and this, and the other two are disregard because of these reasons. Example A, B, and C. Okay, so make sure you go through that, justify that before you hand it in. For your first draft, get someone to read it for you, okay? Because it's important that your stuff flows logically, okay? And then finally, your reference list, okay? And I touched on in-text referencing before, all right? But you need to make sure you're using APA format. Okay. Firstly, in-text referencing, you need to have it, okay? So author and date. Okay, follow whenever you present unique information. Remember, you have this much credibility as an academic. Okay, as do I. Okay, so how do we get our credibility? We do it by actually putting reference sources in. Okay, so for this in year 10 level, I would be expecting an absolute minimum of 6. Okay, so if you look at sections 1, 2, and 3, introduction, where are they again? Introduction, an explanation of recessive dominant genes, and a description of PRA. If you do two references for each of those, then you're very easily at 6. Okay. If you're planning to wanting to be getting good marks in this, I'd be hoping for more than that to show that you're well read, that you've looked around, you've researched the topic extensively, and you've done more than that. So your 6 might be up to 10. Okay, so it's important you do that following APA format. I use the document in Word, and I might put a video of that explaining that up a little later. Okay, but however you do it, make sure you take your time to you're doing your referencing properly in APA format, and if you're not sure, please ask. I hope you find this useful, and good luck with your assignment.